Today's webinar is the first one of a series of webinars, and there will be more webinars happening each Wednesday in October and around the topic of open science, of open access and open science, and more will happen on each Wednesday in October, and then culminate in a panel discussion on Friday, the 29th of October, which will also mark the end of Open Access Week. In today's webinar, we will mainly focus on data sharing and data submission. Next slide, please. But first, let's clarify what open access actually means. So open access means that scientific information is made available online and free of charge to the end user. And this information includes biological and life science data, such as, for example, protein sequences and genome sequences, but also imaging data, experimental protocols, and research publications. Next slide, please. Thank you. EMBL-EBI actually offers a wide range of open access data, and this illustration here presents all the data which is accessible through EMBL-EBI. And you can see that this covers the whole scope of life science, beginning with genes and genome data. We also have protein sequence and protein structural data. There are also databases which refer to chemical biology and databases which refer to whole molecular systems. And we also have databases which are related to literature services and ontologies. Next slide, please. So where does all the data come from? Well, the data comes from you. So this is the scientists, the researchers out there. We, EBI, we really rely on researchers submitting the data to us so that we can archive the data, we can classify it. We might even analyze the data and develop new web tools. And you can use these web tools and the data to generate new insights and to generate new data, which you then can submit to EBI again. You see that sharing is a really central aspect of this data lifecycle. And this is where the EBI data resources come in again. So several of our resources, they accept direct submissions. And on this slide on the right, you can see these EMBL-EBI data resources again. Next slide, please. And with this, I would like to introduce you to our speakers of today, Tony Burdett and Birgit Meldal. So Tony Burdett, he is a technical team leader at EMBL-EBI for archival infrastructure and technology. His team develops services and technology which support activities like data submission and data storage. Today, he will not only talk about open data, but also about fair data. And he will also talk about the impact that GDPR can have on data sharing. We will not touch on the GDPR topic in all detail. We will only touch on it, but if you should have follow-up questions, more detailed follow-up questions about this topic, Tony is very happy to accept these questions after the webinar. Last but not least, Tony will also talk about the importance of metadata and standards for data sharing. I would also like to include you to Birgit Meldal. She's a scientific database curator at EMBL-EBI. She is in charge of databases related to molecular interactions and complexes. And she will talk today about data submission to Intact and Complex Portal. And she will also discuss challenges and benefits of offering open access data. And with this, I would like to say that I'm very happy that both are here today, and I will hand over to Tony.
Okay, thanks, Alex. So I'll jump straight in. So yeah, I think as Alex said, today I'm going to talk about open access data and fair data management, really, which covers a lot of the context that Alex just highlighted about submissions. I'll talk a little bit about the biosamples database, which is one of the resources that my teams run, as well as Fair Plus. I'll very quickly touch on that, which is one of the sort of big projects that we're involved in at the minute about open access and good practices for fair data management. So fair and open are terms that are often sort of conflated or stuck together. For people that haven't heard of fair or the fair data principles, when we talk about fair, we really mean this set of principles that are designed to enhance the value of digital resources, and FAIR stands for findable, accessible, interoperable, and reusable. There's a good paper that you could go read about the FAIR data principles back from 2016. Since they were originally sort of codified, these principles, they've really taken off, and over the last 5 years, I think funders, funding agencies, databases, researchers in general all sort of have really rallied around this and adopted these kind of set of principles as a way to make our data more freely available, more open. But of course, fair data isn't the same as open data. So this is a great definition from the Go Fair initiative. The way they frame this is to say that the A in fair stands for accessible, but under well-defined conditions. So that doesn't mean that it has to be sort of freely available on the internet, totally open. Fair data could be within a company, it could be within a hospital, it can refer to data that's designed to protect personal privacy or some sort of security or competitiveness within the industry context. But it should be accessible as long as you meet the conditions, as well as then these kind of other attributes: findable, interoperable, reusable. And of course, open data isn't necessarily fair either. So you can have data that you put freely available on the web, but without good metadata or without following standards, it's not really findable, certainly not interoperable. So these two things are not quite the same, but we often consider them together because the fair data principles give us quite a good framework for thinking about how that works. And also a great framework for thinking about how important it is to adopt good, open, fair data practices. So we know, for example, that fair data is really important. We know that data science are core elements of health research, innovation, biopharma research, just good like biological and computational biology science. We also know that impact of artificial intelligence and machine learning is growing, and this needs data that computers can understand, not just people from reading a publication, but actually machines can reuse data. But the data generation efforts that we currently adopt tend not to support that level of reuse. And what we know is that data scientists, computational biologists, bioinformaticians spend a huge proportion of their time actually on sort of cleaning tasks. So you can see from this pie chart that sort of, which is one survey on this by Forbes, it is that 80% of data scientists or bioinformaticians' time is spent actually just collecting and cleaning the data. That's a huge amount of time. It's also the thing that data scientists like doing least. They like to get into novel findings, not just data cleanup. And to go along with that, I think the importance of fair and open data is we now know that there's more data resources than ever before. Like data sharing, data generation has a huge impact on genomics and bioinformatics research. We know that GDPR has had quite an impact on how people share their data, what sorts of data they can share, especially data that comes from human subjects, how it's stored, how it's shared, how it's protected. And what we see is that over time, combining data from multiple different repositories, different sources of data, different archives, data from hospitals with data from like the lab, those kind of federated data repositories and combining data for analysis is increasingly common. But what we think is really important is actually practical advice on how to achieve this kind of data combination and how to share your data openly and fairly in a way that means other people can exploit it. So thinking a little bit about how that practically happens, I've got this one slide that describes the data a sort of high-level overview of a data generation lifecycle. And this is coming from sort of the EBI perspective in the context of my team, which deals with a lot of submitted data, a lot of incoming data to some of the big EBI archives. And generally what we see is these kind of broadly 5 steps of a life cycle of data generation or a data generating project in bioinformatics. And that starts with things like planning, so metadata and data planning. What standards should we adopt? Does everyone agree within our project to use these kind of standards of data and metadata? And how will we sort of then go about collecting it? Moves into this second phase. So what's the format? Does everyone understand it? Can we collect data in a way that meets our internal standards? Usually there's then some sort of storage component. So as you start to carry out assays, whether that's sequencing data or imaging data or whatever else sort of types of technical assays you're trying to do, there's generally some sort of, within your project, there's some sort of general place where you store and upload your data. Quite often then we go through a validation cycle. So is all of our data valid against some standard? Are the experimental designs, the protocols that we use to collect this data consistent? What file formats do we have? And finally, you go through this brokering stage, which is generally sort of like the publishing of the data. So often it's coupled to when you write your publication, you're going to release your metadata and your data, release those files to some sort of final location. And this is the kind of sharing process. And what that looks like, if you plot this out over time with the amount of data management effort that people tend to put in, is you start down on the bottom left here. And what happens typically is the sort of geekiest person in a lab who knows how to do this stuff sets up data as some sort of data management system, probably in Excel. You start with a spreadsheet, you start bashing some data in and stick all the files on a directory somewhere. You then go through a phase where you kind of collect the samples, maybe from a biobank or maybe from recruiting them directly, or if you're in a more clinical setting, maybe you sort of recruit the patients. And you then go about this kind of data collection. So you do 3 years of data generation and sort of study, assaying like more and more samples to devise your experiment. And then you get to the end and you publish your paper, and then the journal says you've got to comply with some community standards. We want you to make your data publicly available. We want you to share it openly. At which point you have to then sort of talk to like one of us at the EBI, a data submission resource, and we say, oh well, you should adopt these standards. You should upload it here. Here's how we validate it, and here's how we want you to submit. And you have to do all this work again, all this kind of management, planning, storage, collection. You have to redo it all in accordance with standards that are sort of dictated by major archives. And that's really, really painful. And so what we see is you feel this pain of data sharing, where I've shown a biobank on the left-hand side of this diagram. It could be anything, so sort of wherever you collected your samples from. And EBI and other similar archives on the right-hand side, and poor researchers are happily doing their experiment stuck in the middle. And then we say, okay, well, now you need to submit data to EBI archives. And straight away, you feel the pain of having to reformat your data. And I'm sure there's plenty of people on the call who've been through this process in the past. We tell you how to lay it out, and we tell you all the sort of different types of vocabularies that you should use to annotate the data. And practically what happens is you go through a really painful copy-paste process, taking data out of one format into another. Often it's a spreadsheet or it's Excel, and you find this phenomenon of it's a known phenomenon that like there are many errors that are introduced by just this data reformatting or metadata reformatting process. You get gene name errors that are widespread in the scientific literature, and crazy things happen where gene symbols like Sep2 are converted to the 2nd of September, and they started renaming the gene symbols now to avoid these types of collisions because it's easier than changing people's behavior. This it's a known phenomenon. And actually, what we try to encourage people to do is to think about a sort of slightly more stepwise and incremental process to open data sharing. So rather than holding on to your data and setting it up in those kind of spreadsheets or bespoke lab specific structures, what we encourage people to do is try and adopt shared community standards at the start, close to the start of their project. And then as you go through this kind of data sample collection, data generation process, you can start to use existing data resources, things like the biosamples database as you collect your samples, for example. And then as you generate the data, you sort of upload it, you're continuously validating the data that you're generating. And then by the time it actually comes to publication, you get this kind of one-click possibility where you can simply hit the button, make all the data that you've generated over the last 3 years public in one batch, but it's all been generated against community standards and validated throughout. And this is kind of our sort of recommendation for how to adopt good open data sharing practices. And there's I want to dive a little bit deeper into that now and actually think about, well, if you start doing this, how does this actually happen? And what we see within EBI archives in general, there's some general trends here, is that submitters to EBI resources tend to fall into one of three categories. You've got sort of someone who's more like a bench biologist or a one-time submitter who maybe has written the study. Perhaps they're a relatively junior person in the lab. They need an accession number because the journal says so, and they want to do the simple, logical, easy submissions. You then have slightly more frequent submitters who perhaps have matured those kind of data sharing infrastructures as sort of adopting something that looks a little bit more like that good data sharing practice and know how to do their data management, but have these kind of niche requirements like what sort of what version of the ontology should I use, and can I adopt your validation so that I can validate it before I send it to you? And then there's power users who are generally streaming data to us directly. So this might include some of the big sequencing centers, for example. And for those scenarios, they need to avoid any manual intervention. And you get this kind of curious phenomenon if you start to map those types of submitters out, which I've called sort of novice, expert, and power submitters here, where what you see is that most of the people that submit data to EBI archives are novice submitters, by which I mean they will only ever do one or two submissions. But most of the data, when you look at it by volume of submissions, 80% of our submissions come from expert or power submissions. So we get really high volumes of data being streamed through from large scale submitters, and then lots and lots of submitters that only ever submit one experiment. And we really need to sort of target our solutions to those two different types of submitters separately, and it requires two different styles of data management. And just to build on that, you can see these trends are echoed across the different archives. So here I've cited ENA, EGA, and Array Express. ENA is even more skewed towards power submitters. Most of the data in ENA is streamed through from direct sequencing centers. And just to build on this idea about coordinating open data sharing with EBI archives, there's quite a lot of life cycle that happens within the different types of archives. So I talked about it at the start, where you have sort of samples being recruited from a project and dispatched before you've perhaps done the sequencing or done some further downstream analysis. And the biosamples database is a good example of how you can start to approach this strategy of like breaking up some of this kind of coordination types of activity. So the biosamples database is an archive of information about biological materials, and we store sample data from both internal, by which I mean sort of other EBI archives, and external sources, by which I mean directly from submitters. So in other words, if you've started a data intensive project and you know that you're going to be generating sequence data or variation data from those samples that you've collected, and you want to pre-register your samples in biosamples, we can give you the accession back so that you can link submissions together. In other words, you can say, well, I have sequence that's gone to ENA, I've done a functional transcriptomics experiment, and I want to put that data in Array Express, and I've got the literature that's in the paper that I published in Europe PubMed Central, and I want to connect that across these kind of, here are the shared samples. And biosamples will give you the ability to do that. And there's now 18 million sample records in biosamples as of September 2021. And I think the important thing to focus on here is that data, especially open data, has little value without good metadata. So by starting to sort of create a single source of sample descriptions, you can apply this trend more widely, but here for biosamples, you start to build up this picture where you can say, well, we can reliably use good standards, we can validate against those standards, and we can start to answer some specific use cases. And some of these are drawn from the Global Alliance for Genomics and Health. So if you start to ask questions like, I want all of the data that come from samples from male individuals, or I want all of the data sets for which individuals have consented, according to some ethical consent code, or I want all individuals that originated from a specific geographical location. And I can find those by looking at the samples where they were recruited, and then look at the links to the underlying data set, supporting this kind of findability and interoperability components of the FAIR data principles. And one really good example of where we do this is in the COVID data portal. So me and my colleagues at EBI have been heavily involved in the COVID data portal. I think Birgit will talk a little bit more about this later on. And we see some specific examples. So essentially, we see researchers coming along wanting to find all the COVID-related samples, and then all of the data that may have been derived from those samples. And there's a bunch of different ways to describe COVID samples, and you can see some of the examples on the left-hand side here. And we see some common challenges. They can be curation challenges, but earlier adoption of samples helps us avoid the need to do retrospective curation. And these are things like looking at special characters, looking at acronyms, typos, using the right ontologies. These things are all really, really valuable and help us do this kind of data discoverability or findability of COVID data, and then powers this kind of open sharing and reuse scenarios. And this works in practice. We now know, based on the work that we've done, the standards that we've adopted, and the encouragement of people to share their data openly, we now know that there's 1.5 million samples in the COVID data platform with data associated to them. And you can go look at the COVID data portal and see some of these details, and they all have very nicely well harmonized attributes that describe the characteristics of those samples, where they came from, sort of the severity of the disease, these kind of things. So that's nearly the end of my presentation. I wanted to finish with a little bit of like practical advice. So I mentioned the FAIR Plus project right at the start, and what we do within FAIR Plus is talk about fairification. I won't touch on this. I'm happy to take questions on it at the end. But what we advocate is thinking about your data requirements, the resources that you need, and the capabilities that you have to do this kind of good, fair data management early, and then incrementally work on it to improve. Very happy to take questions on this later. So just to finish off, I wanted to summarize by saying that the sort of overview of my presentation here is that fair and open data is vital to research, but we know that the costs are high when you can't reuse data. There's a huge opportunity cost from having to regenerate data that already exists. Fair and open data aren't the same. They're subtly different. EBI archives provide support for lots of this open data sharing, so you don't need to go do it all yourselves. You don't need to reinvent the wheel. But engaging early in your projects is a good way to make it simpler and easier, especially if you adopt some of these fair by design strategies. And finally, there's loads of resources available providing guidance about how to be more fair, how to do open data sharing, and that includes FAIR Plus. But actually learning from case studies and see what others have done, how to facilitate open data sharing is a really valuable approach. I think Birgit's going to talk a little bit more about some of those case studies that we see in a second. Very quick acknowledgement slide. This is my team, the Archive Infrastructure and Technology team at EBI, some of our funders, which I should acknowledge as well. So I think with that, I'll hand over to Birgit.
Okay, so thank you very much, Tony. That was an excellent introduction. I will share some stories with you to explain a bit more the advantages of open data and how we make data fair, and how that all helps us with our interoperability between resources and our exchanges with our users. So I'm a curator for Complex Portal, which is part of the molecular interactions team that also includes Intact, and I will be covering both of these resources in my presentation. So the databases are both manually curated data from publications or pre-published papers. I'll come back to that later. We use community standards. Everything is free to access, download, and if you wish so, you can even install the whole database in your locality. Up until recently, the data had a CC BY license and Apache 2 for the software, and I'll come back to that at the last part of my presentation, which meant that at the surface, these data resources were both fair and open. A quick introduction to the two resources. Intact is a deposition database for experimentally derived molecular interactions data. We curate directly from published data, and we very much encourage direct submissions during the publication process so that we can curate the data and have a conversation with our submitters to make sure the data is correctly in the database. And often we can actually capture more metadata than the authors will be able to provide in supplementary materials. The complex portal is a knowledge base that unifies experimental information about molecular complexes. And these complexes are defined as the functional biological units of the cell. All of our curation in complex portal is also based on published evidence. We don't accept experimental data that would go into Intact, but we accept and encourage user requests for updates to existing entries or curation requests for new complexes. Couple of words about the data flow. It's slightly different between Intact, which is the deposition database, and complex portal, which is the knowledge base. So in Intact, as I mentioned, the data input is either from a already published paper or through direct submissions. More about that later. And then periodically through our roughly bi-monthly releases, the data goes out to our website, to our web service, and is accessible through the FDP, and to further to federated access points, Imex and Psychic, and I will talk a bit more about those later. A lot of other tertiary resources access our data. A lot of tools like Bioconductor and our packages can access our data. And one of those, for example, is the Intact Cytoscape app that we released earlier this year. And finally, a lot of our data is reciprocally cross-referenced in other resources. So for proteins, for example, we use UniProt as the primary reference database, and in return, UniProt provides cross-references to interactions that the particular protein is involved in on the protein pages. Intact can also use complex portal identifiers as members of interaction, and complex portal uses interaction evidences from Intact as evidences for complexes. We reciprocally exchange gene ontology annotations. Reactome captures our data, and you can overlay interactions over reactions and pathways. And for example, OmniPath uses Intact for their experimental referencing of the signaling pathways. And there are many more applications. This is just an example, and there are probably a lot of tools that we don't even know of that use our data, but they use it because they can access it through the open access pathways that we provide. Complex portal is slightly different. Again, as I mentioned, all of our data is based on experimental evidences. These are usually described in publications, and we often draw directly from the deposition databases such as PDB or Intact with the Imex partners. More about that in a minute. We reference out to PubMed and to PDB, but at the moment they don't provide cross-references back to complex portals, so that's why the color of the arrows is slightly different. On the left-hand side, as I already mentioned, there's reciprocal use of data, both as annotation objects and cross-referencing between complex portal and UniProt, just like Intact does, and between complex portal and Intact and other Imex resources. We also exchange gene ontology annotations for complexes with the Go consortium, and we cross-reference to complex instances in other databases such as MatrixDB, Campbell, or Reactome, who then cross-reference back to us. All of this is only possible because the data is fully accessible. And finally, there are a number of databases or other data resources that take our data and reference back to us, such as SGD, the Saccharomyces genome database, and the genome properties, wiki pathways, and wiki data repositories, which I'll come to in a moment. How do you submit to Intact or Complex Portal as an example for a deposition or knowledge base? We try to make this as easy as possible for you. So for Intact, on our data submission website, you can download an Excel template and fill in all the data that's required. There's a minimum requirement of what data we need. You fill in the Excel sheet and then you click on Contact Us, which opens up a contact form, a standard EBI contact form, attach the Excel data, and it gets sent to the Intact help desk. And then we will have interchange with you as the author to make sure we get all the data correctly into the Intact website, into the Intact environment. Depending on the size and extensivity of extent of the data that you're providing, there might be a lot of communication. We might ask you for more metadata if that's available. And in some cases, we can also share the file for you to check before it goes into the release process. I should mention, if you submit the data pre-publication, we keep it confidential until either you let us know that it should be part of our next release, or we notice that the publication has come out, and then we will add the data to our next release. You can also request curation of publications that are already released if you need these for your downstream analysis. So just send us, again, through the contact form, the PubMed ID for that publication, and we will incorporate it into the next release. Complex Portal is even easier. You just have to hit the yellow tile on the homepage. It opens the equivalent form that gets sent to Complex Portal help desk. All we ask you to is send us a recommended name of the complex, a list of components of the complex, ideally with Uniprot identifiers, as that makes it specific to what we're talking about, and some kind of reference, some kind of PubMed ID. You can give us more information, such as if this complex has already got a structure in PDB or EMDB or any other helpful extra information. And then we will curate the complex from there. Okay, so in the rest of my presentation, I will tell 3 stories about how we made use of open access and FAIR. And the last story is a little, it comes a little surprise. I won't give it away yet. So the first story is about how we built our community. In the late 1990s and early 2000s, a number of different molecular interaction databases were established. They all established independently of each other with different topic focus points. And it quickly became clear that it wasn't sustainable to run all of these databases independently. For starters, it was a real headache for the user because they had to access each database separately, download the data, do whatever mapping was required, because every database had started off with their own data model, their own standards, their own formats. So it was a real headache. So in the early noughties, the community came together and decided, well, the least we can do is come up with a common standard. And this common standard was the Proteomics Standard Initiative for Molecular Interactions, or short PSIMI standard. And from then on, the majority of molecular interactions databases followed this standard, which meant the users could download the data from each of the resources and amalgamate it at their end. Of course, that was still a headache for them. And then we also provided a federated interface, which is called Psychic. I haven't got much time to explain to talk about this, but essentially any resource that could provide data in PSIMI standard could set up a Psychic service, and the user could was only required to make one search, and from this search they could get the data from all of the resources. But they still had to download a separate file for each resource, but it was one, or it's still existing. It is one interface, and it can also be used programmatically. So from there on, the community was set up in terms of standards, but the curation was still independent. And it meant that the different curators were often picking the same papers to curate. So even though the users were able to access the data through Psychic, through a single search, they still had to pass through the data and make sure they ended up with a non-redundant data set. So in 2005, the Imex consortium was established to federate the curation as well. The databases were still independent databases, but we built a tool that allowed each curator to check whether the paper they wanted to curate was already curated in one of the partner databases, and it would block the curation of duplicated entries. So all the data that's available through the Imex consortium is manually curated, experimentally based, and non-redundant data. And this was a huge improvement for the users. In the intervening years, most of the work that we've done was, apart from curating more content, updating the standards and formats to account for new and changing data types. So we had to stay with the times. But also another issue that faces many data resources is that you have to streamline them, because funding is always an issue. So because we had the community standards in place, we were able to eventually integrate the curation and the backend of all the Imex partners into the Intact database. The first database that was incorporated into Intact was actually one that completely lost their funding. And because the data in that database complied with the PSI-MI standards, it was a fairly straightforward import operation, and the data is still there available today because it was standardized. It was fair and is still fair. And yes, over time, all the Imex partners decided to move over and use the Intact environment, not just for the standards, but also for the actual curation and the backend. The other advantage of that was that now the entire Imex the Imex compendium, the whole data from Imex, gets released at the same time. And whilst previously the user had to could do a single search for Imex related data, they still had to download individual files from each partner through the Imex website. Now there is just a single search and a single file because all the data is federated through the Intact environment. So that's a story about Intact. And we couldn't do this without the data being open, and our users wouldn't have access to this federated, and standardized data without it being open access. So the second story is about how the Imex environment provided us with a way of curating over the last 18 months. I don't have to explain much more about what happened at the early 2020, but it's incredible how quickly COVID-related, SARS-CoV-2-related data was available. And it was only available because everyone agreed that this data was so important that it should be made open access immediately. So the sequence was then January the 1st, structures were released in February. And by March, we had the first 2 SARS-CoV-2 human interactomes available. At that point, we just started working from home, and it was about the second week of working from home that both Intact and Complex Portal curators, in fact, all Imex curators started to curate coronavirus-related data. Moreover, because we're curating based on standards, we also relied on standard identifiers for the new proteins. So UniProt provided us and the whole community with an emergency release, or through an emergency release, with the new UniProt identifiers. UniProt skipped their regular 8-week delay to get a release and provided this immediately to everyone, which meant by April, Intact and Complex Portal were able to release the first coronavirus-related data, interaction data and complex data. And this was a whole Imex effort on the interaction side. We then put all that data together and ran some analysis and deposited this data in bioRxiv in June. If it wasn't for pre-publication service, we wouldn't have been able to get this publication out that quickly. And by the time, 3 months after we started curating, we'd already duplicated the amount of interaction data in the database. A third major interaction was released just after we've done our analysis for the initial bioRxiv deposition. And by December, so about 6 months after our bioRxiv submission, we finally had the peer-reviewed paper out. So through the open data availability, or through the access of open data, open access through bioRxiv, we were able to actually tell the community our data was out there, and this is what you have 6 months earlier than we could have done through the whole review process. By that point, the data had doubled again. We were now at nearly 4,500 interaction evidences for coronavirus data. This was more than just SARS-CoV-2. This was SARS-CoV and other coronavirus data as well, and related human and other experimental data. And a huge advantage at this point was that most publishers had agreed in the early 2020 that any publication that was related to the pandemic would be immediately open access. So they've done away with the paywalls. This was obviously incredibly important for the community, for researchers who maybe didn't have access to certain journals or would have had to pay a lot of money. For us, it was also much quicker because we didn't have to go through library requests. And again, it was saving us the money for the library requests. So we were able to curate this data much, much quicker. And just to put you at ease, any of the curations we've done from bioRxiv-based depositions, we regularly check if they've now been peer-reviewed and we update the data. For any other curations in Intact, we only curate the peer-reviewed version simply because it saves time and it makes it more stable, the data that we put out. So after that, and still to this day, we're still always checking for updates to the COVID related data. So now to the last story. So there was a little side story that I was personally involved in as the complex portal curator. And it opened the question of how open is open access. When we released our first coronavirus release in April, we tweeted about it, and someone I'd met at the Biocuration conference a few years earlier from the Wikidata community contacted me and said, "Hey Birgit, are you going to join the Biohackathon this week?" I'm like, "Well, I'm not a developer, I'm a curator." And they said, no, we do need people who really know about the data, and we do want to bring the complexes into Wikidata because there's a huge semantic data community who was struggling to access the data from the way we were providing it, the formats that we were providing. So they were really interested in getting these complexes in. And there have been a few complexes that have been manually curated by Wikipathways curators and cross-linked to Complex Portal, but we're talking about a dozen. So by the end of the Biohackathon, we created Wikidata entries for the 11 SARS-CoV-2 complexes that we just released. But we had to go through a semi-automatic process for this. So there was a lot of manual mapping and data checking involved, which is normal for the start of any kind of new pipeline. But we knew this wasn't sustainable in the long term. We needed to develop a pipeline, which is a Wikidata bot. So just as a side note, because I was in the Biohackathon and there were lots and lots of tracks going on, I was also able to advise the knowledge graph track of how to access the Intact and Imex molecular interactions data, which they then brought into the knowledge graph. So like Tony said, there was a lot of effort made in providing interoperable COVID-related data access points. And of course, the COVID-19 data portal is one of the biggest. And all of this, like the knowledge graph, they all feed into that. So the next sort of 9 months, we slowly worked away through developing this bot and working on how best we can actually bring complex portal data into Wikidata. I had various video calls and a Slack channel, and there was a small group that was spread across half the world really worked on it. We got again together in a hackathon in December, and at that point we finally managed to get the first version of the bot going, and that was bringing in the rest of the coronavirus related complexes. And then it took another 6 months until we were at the point where the bot could run across the entire complex portal compendium. So you're wondering why that took so long. Well, Wikidata is a community project, and some people are actually working on it in their spare time. Others can incorporate it into their normal projects, but everyone's running different things at the same time. We were then held up by the Wikibot approval process. So there was a user who was worried about how frequently we were referencing back to the original source of the data. So Wikidata allows you to add a reference to each entity or each element that you put into Wikidata. And so we finally satisfied the user, but we had to make some tweaks in the bot, and there were technical issues which we don't have time for now. We finally managed to get there, but we also had to jump through the licensing hurdle, and this link brings me to the last slide. I'm not an expert in licensing. I relied on project collaborators who looked into this, who were using Wikidata a lot. So the prerequisite for using Wikidata or linking things in Wikidata means that every element in a Wikidata entry has to have their own Wikidata entry. So if you populate a complex portal, a complex entry in Wikidata, and you want to list, say, the component proteins of the complex, you need to have a Wikidata entry for those proteins. So they already need to exist somewhere. And there was already a bot from another project that was creating most UniProt proteins in Wikidata. So that was okay. And also for most gene ontology terms, they were already present from a previous project. However, an automatic Wikidata bot can only ingest data from CC0 resources. CC0 means reuse without the requirement of giving the credit of where the data comes from. So you can freely reuse anything in any way. Now, as I mentioned, Wikidata can include references, and being academic scientists, we were very keen to use the referencing option, which is there in every field in our entries. In terms of the license for complex portal, this wasn't a big thing because we had the environment to actually decide there's nothing to us stopping moving complex portal to CC0. And that was done fairly quickly. But we're still having a bit of an issue, and we haven't solved this yet. There are some entities in complexes that come from non-CC0 resources. So there are complex members that are not proteins, so for example, RNAs that are linked to a resource that is not CC0 yet. So whilst we could technically create these entry pages, these entity pages for those other elements in Wikidata through the bot, we're not allowed to do this because the resource isn't CC0. We also discovered that whilst the protein box bot that creates UniProt entries creates canonical UniProt entries, it doesn't create entries for the isoform and protein identifiers, which we use sometimes in complexes. So the short-term solution will be to, again, go manual and add manually these other data, these sort of missing bits of data. The data resources are open, they're CC BY, so it doesn't stop us from manually from manually adding these entities. But as I mentioned, this is non-sustainable. The whole idea of this is that we automatize everything and run the bot with every release of complex portal. So at this point in time, at the moment, Wikidata is bringing in all the data from complex portal and all the cross-referencing, where the entities are already created in Wikidata prior to the bot running, or they come from CC0 resources. but any CC BY resources, even though they're open, can't be brought in. So they're currently missing. So on this point, I think Alex will summarize things. And I know it's a little bit of like a negative point to finish with, but it's also a cautionary tale to see how different types of open data aren't the same. So thanks on Alex. Oh yeah, of course I need to thank everyone, but we're running out of time to go through the whole long list. So over to Alex to summarize.
Yeah, thank you very much, Tony and Birgit. I just really want to very quickly add a few thoughts or summary thoughts to this as take-home messages and to emphasize the importance of these ideas. So it has been raised several times during both talks that data sharing is important. And just to summarize some of the reasons for it. So data sharing and making data open increases the visibility of research data and information, as we have seen. And of course, this also can help to make quicker collaborations much more quicker and efficiently. Also, data sharing greatly accelerates research across the life sciences, and open data and data sharing enables scientists to quickly react to and build upon existing research, as we have seen in this talk. And last but not least, data sharing increases scientific reliability, reproducibility, and traceability. Of course, this list is not complete, and you might think of more benefits. If you could please go to the next slide. If you need further guidance on how to submit data to EBI, I would like to refer you to our data submission webpage, which the link for this is also given here. So you will also find a questionnaire there, and this questionnaire will ask you several questions about the nature of your data set, and thus this will help you, the questionnaire will help you to narrow down a database which is suitable for your data set, if you should not know to which database you can submit your data set to. I would also like to encourage you at the end of this webinar to keep in mind that you should plan your submissions as early as possible in your research projects. And whenever you have questions or get stuck, please reach out to the people who work at the databases, reach out to the biocurators, because they are very likely to help you. Next slide, please. And if you have further questions, there are help desk at the data resources you can contact. And I would also like to point you to our online tutorial, which is called Bringing Data to Life and Data Management for the Biomolecular Sciences, where you will find other content or more content around the topics of open access and data sharing. And of course, please keep an eye on our upcoming webinars. Next slide, please. So our upcoming webinars are accessible through our EBI training webpage. And as I've already mentioned, we also record all our webinars, and the recordings are also available on our EBI training webpage. I would very quickly like to remind you that there is a survey that will be launched, feedback survey at the very end of the webinar. And I would like to ask you to answer this. And now we can use the last minutes to answer some of the questions that were actually submitted by our attendees. So let's start with the first one. How do you foresee the clinical data sharing considering sensitive privacy issues?
So I guess I can make a start. Birgit, feel free to chip in. This is a really tricky, really interesting question. I think there is a move towards overall with under sort of like the Global Alliance for Genomics and Health umbrella and elsewhere, there's a move towards more shared standards, but privately held data. So whether that's in national centers or hospitals. So the European Genome Phenome Archive specifically has this kind of federated EGA approach, which is designed to ensure that, like, within Europe, different countries can have different nodes of the database and therefore can retain data privately, which you need to go through a process of requesting access to. And this is fair without necessarily being open. You know, we talk about sort of managed access and controlled access, but actually making sure that everyone adopts the same standards and the same procedures for sharing data makes that data fair. It is a complicated issue. I think it's an ongoing discussion, yeah, I think it will and continues to be a challenge. But I also think EBI are sort of involved in a lot of initiatives that are leading the way, and there are concrete recommendations that are coming out that perhaps is a bit too broad to cover in the last 2 minutes, but I think would be an interesting topic to follow up on.
Yeah, I don't think I've got anything to add because we don't actually get clinical data in our resources.
Yeah, perfect. Now then let's move on to the next question. Asking as a support scientist, which backend is employed by Intact /EBI? IRODS? If so, are IRODS federations supported for data retrieval? Can any one of you answer this question?
I don't know what IRODS are, certainly not what we're using. Tony, do you know?
Yes, but I don't think I know enough about the way this works within other resources to answer the question. So perhaps, Christian, we could ask you to drop, like me and Birgit, an email offline afterwards, and we can dive into the details or find out the answer for you.
Yeah, so our backend at the moment is a relational database and a graph database, and one of the two or both is the case for most of our resources, I think. But yeah, it is resource dependent.
Okay, thank you. Margaret asks, are data submission policies and procedures identical among EBI, NCBI, and DDGB? Personal experience tells me they aren't, but...
Yeah, this is definitely one for me. So it is dependent on resource. It's resource by resource. Some resources have natural partners across EBI, NCBI, and DDBJ repositories. So the obvious one is the INSDC collaboration, which is International Nucleotide Sequence Database, of which ENA is a part, and as well as SRA, NCBI, and DDBJ. The procedures and processes are closer there. They're not identical, but we try to keep them as close as we can. If you think about things like EGA, they often are identical. So there's a submission resource at EBI and at Barcelona for EGA that have really, really closely aligned standard operating procedures that strive for being identical. Whereas there are other databases that sort of operate in completely different spheres. So the biostudies database at EBI versus bioprojects at NCBI don't overlap and don't exchange data at all. So it's a bit resource by resource. We talk to our colleagues at NCBI and DDBJ a lot, and we try to align, but they're not always identical.
Maybe one addition. So the Imex consortium has solved that issue by actually providing the same import for everyone. So there's only any interaction, molecular interaction data goes through the same pipeline, and it gets picked up by whichever database decides, whichever curator decides to pick that paper.
Wonderful. We have only 1 minute left, but Birgit, you said you would like to answer one question live. Maybe we can do that very quickly, the one from Christian. Does Intact EBI support metadata standards for virtual ligand screening results too? Seems not to be in the list.
Well, actually, I was going to say, I'm not entirely sure about the details of that. So if you can hit the feedback button on the Intact website and ask the question to the help desk, then the relevant person can pick that one up. We can deal with pretty much all the data, and we can deal with small molecules if they've got KEBI identities. So it might be possible. And there was another question, I think, to me, how Intact data derived from experiments can be curated without being properly validated? How do you know the data are reliable? It's a very easy answer. If we think a paper, all the experiments in a paper seem a little bit dubious, we just won't curate the paper. But if we think the paper is sound, then we curate all the data, but we don't actually make a judgment on the validity of the data. We curate what's in the, what the authors have seen. There are some, yeah, some bits about high throughput papers. The authors often provide a cutoff of what they think based on their in silico post-processing, what is high quality, low quality data, and then they often say, well, actually only the high quality data based on our author assessment should go into Intact. But again, it's the author who assesses what should go in.
Okay, thank you. We are at the end of this webinar. I would like to thank Tony and Birgit a lot for being here and for this great discussion. And I would also like to thank the attendees, and I hope to see some of you again for some of the following webinars on open access. Thank you and bye.
Thank you, everyone.